Previously in Part 1.
In our last video, we broke down the master blueprint of life's ultimate shield, the Fluid Mosaic Model. We saw how every cell wraps itself in a fluid, shifting ocean of lipids and proteins just to stay alive, but a blueprint is only as good as the materials you use to build it. Think about it, if the membrane is essentially made of fluid oil, what stops it from literally melting when a cell gets too hot? Or freezing into solid ice when the temperature drops? To survive a chaotic world, Prokaryotes and Eukaryotes had to engineer incredible chemical reinforcements. Today, we are moving past the basic blueprint to look at the hidden molecular upgrades that keep these membranes from self-destructing. Welcome to Part 2. Let's start with the battle of the ultimate cellular thermostats, cholesterol versus Hopanoids. Let's look at the eukaryotic membrane first. Animals and plants live in highly shifting environments. If the temperature gets too hot, the fluid membrane could literally disintegrate. If it gets too cold, it turns into a rigid, frozen solid, killing the cell. To solve this, eukaryotes engineered a master thermostat molecule, STOLS. Animal cells, this is cholesterol. Yes, the same cholesterol your doctor warns you about is actually a structural lifesaver at the cellular level. When it gets hot, cholesterol grabs the phospholipids and pulls them together, keeping the membrane intact. When it gets freezing cold, cholesterol wedgily inserts itself between the tails, preventing them from packing too tightly and turning into ice. But what about bacteria? They don't make cholesterol. In fact, if you see cholesterol in a bacterial membrane, it usually means that bacterium is a parasite stealing it from a human host. Instead, bacteria use an ancient molecular cousin called Hopanoids. Hopanoids have 5 rings instead of cholesterol's 4, making them slightly bulkier and incredibly robust. They perform the exact same job, stabilizing the membrane across wild temperature shifts. Because bacteria have been using Hopanoids for billions of years, these molecules are highly stable. When bacteria die and sink to the bottom of the ocean, their Hopanoids turn into fossils. Geologists actually use fossilized Hopanoids in ancient rocks to trace the history of early bacterial life on Earth. Now we get to the biggest, most mind-blowing difference between these two systems: energy production. Every living cell needs to generate ATP, the universal currency of cellular energy. To do this, cells have to pump protons across a membrane to charge it up like a battery, and then let those protons rush back through a spinning molecular turbine called ATP synthase. Because eukaryotic cells are massive, complex cities, they don't run this dangerous, high-voltage electrical grid on their outer border. Instead, they delegated this job to internal, membrane-bound power plants called mitochondria. The eukaryotic outer plasma membrane is a peaceful zone. It is strictly used for cell signaling, letting nutrients in, and sending waste out. But prokaryotes? They don't have mitochondria. In fact, they don't have any internal membrane compartments at all. This means the bacterial cell membrane is forced to do everything. It doesn't just act as a barrier, it is the power plant. The entire electron transport chain is embedded right there in the outer membrane. A bacterium turns its entire outer edge into a massive, highly charged battery. Imagine if the outer walls of your house were entirely covered in live, exposed high-voltage wiring. That is what a bacterial cell membrane is doing every second of its life, and because it is holding such a massive electrical charge, the bacterial membrane cannot afford to bend, twist, or tear. To protect this fragile electrical barrier from popping under pressure, bacteria coat the outside of their membrane with a rigid, armor-like mesh called a peptidoglycan cell wall. It locks the membrane in a tight, pressurized capsule. Eukaryotic cells rely on flexible membranes to perform endocytosis, actively engulfing large molecules and fluids. In contrast, prokaryotes are encased by a rigid cell wall, which prevents membrane invagination and requires them to transport nutrients and waste exclusively through specialized channels, pores, and active transport proteins. Because the eukaryotic membrane is free from the burden of generating energy, it can do something truly spectacular. It can change its shape. Eukaryotic cells can perform endocytosis and exocytosis. If an immune cell encounters a virus, its membrane fluidly bends inward, wraps completely around the invader, and swallows it whole. It creates custom-made bubbles called vesicles to transport bulk materials across the border. Prokaryotes can almost never do this. Because their cell membrane is holding a massive electrical charge, it cannot afford to widely bend, twist, or tear. To protect this fragile electrical barrier, bacteria coat the outside of their membrane with a rigid, armor-like mesh called a peptidoglycan cell wall. This wall keeps the membrane locked in a tight, pressurized capsule. Because of this rigid cage, bacteria cannot swallow things whole. Instead, they must secret enzymes into the environment to digest food into tiny pieces before sucking them through narrow protein channels. Now, if there is one thing you need to know about biology, it's that nature loves to break its own rules. Just when you think you have cell membranes figured out, you meet the Archaea. Archaea are prokaryotes, but they are famous for living in places that would instantly cook, dissolve, or crush any other life form, like pure volcanic acid or boiling deep-sea vents. To survive, they completely re-engineered their membrane chemistry. While bacteria and eukaryotes use ester bonds to attach their lipid tails, Archaea use ultra-strong ether bonds. Even crazier? Many Archaea don't have a bilayer at all. Instead, their lipid tails fuse completely in the middle, turning the membrane into a single, solid lipid monolayer. Think of it like swapping a zipper for a solid steel bar. This prevents their membrane from peeling apart, allowing them to literally swim in boiling water without melting. On the complete opposite end of the spectrum, you have Mycoplasma. Remember how we said bacteria must have a rigid cell wall to protect their membrane battery? Well, Mycoplasma threw that rule out the window. They completely abandoned the cell wall. To survive without armor, these sneaky pathogens steal cholesterol directly from their human hosts, using our own lipids to build a flexible, tough shield that tricks our immune system. When you step back and look at the big picture, the cell membrane is far more than just a passive skin. It is the ultimate evolutionary canvas. Eukaryotes chose flexibility and complexity. By moving their energy grid inside to the mitochondria, their outer membrane was freed up to bend, shapeshift, and build massive, multicellular organisms like plants, animals, and humans. Prokaryotes chose raw efficiency and rugged independence. They turned their simple outer barrier into an all-in-one survival engine, acting as a wall, a filter, and a power plant all at the same time. Ultimately, every single living thing on Earth depends on an oily, microscopic shield just 2 molecules thick. If you made it this far and successfully spotted the extra Hopanoid ring earlier, you are officially a biology master. Hit that like button if you learned something new, make sure to subscribe so you don't miss our next deep dive into the microscopic world, and drop a comment below letting me know which cellular engineering strategy you think is superior. Thanks for watching part 2, and I'll see you in the next video.